Hello, welcome to Biology, the scientific study of life. Fate mapping. In classical studies performed in the 1920, German embryologists Vogt charted fate maps for different regions of early amphibians embryo. Vogt's results were among the earliest indications that the lineage of cells making up the three germ layers created by gastrulation is traceable to cells in the blastula. Before gastrulation has begun, later researchers developed more sophisticated techniques that allow them to mark an individual blastomer during cleavage and then follow the marker as it was distributed to all the mitotic descendants of that cell. Developmental biologists have combined fate mapping studies with experimental manipulation of parts of embryos, in which they study whether a cell's fate can be changed by moving it elsewhere in the embryo. Two important conclusions have emerged. First, in most animals, specific tissues of the older embryo can be attributed to certain early founder cells. Second, as developmental proceeds, as cells' developmental potential, the range of structures that it can give rise to becomes restricted. Establishing cellular asymmetries, the axis of the basic body plan. A bilaterally symmetrical animal has an anterior-posterior axis, a dorsal-ventral axis, and left and right sides. Establishing this basic body plan is first step in morphogenesis and a prerequisite for the development of tissues and organs. In non-amniotic vertebrates, basic instructions for establishing the body axis are set down early during oogenesis or fertilization. For example, in many frogs, the locations of melanin and yolk in the unfertilized egg define the animal and vegetal hemispheres. Respectively, the animal-vegetal axis indirectly determines the anterior-posterior body axis. Fertilization then triggers cortical rotation, which establishes the dorsal-ventral axis and at the same time leads to the appearance of the gray crescent, whose position marks the dorsal side. Once any two axes are established, the third, in this case the left-right axis, is specified by default. Of course, specific molecular mechanisms must then carry out the pattern associated with that axis. And amniotes, the body axis are not fully established until later. That in sex, gravity is apparently involved in establishing the anterior-posterior axis as the egg travels down the oviduct in the hind before being laid. Later, pH differences between the two sides of the blastoderm cells establish the dorsal-ventral axis. If the pH is artificially reversed above and below the blastoderm, the port facing the egg white will turn into the ventral side, and the side facing the yolk will turn into the back dorsal side opposite to their normal fates. In mammals, no polarity is obvious until after cleavage, although recent research suggests that the orientation of the egg and sperm nuclei before their fusion may play a role in determining the axis. A restriction of cellular potency. In many species that have cytoplasmic determinants, only the zygote is totipotent, that is capable of developing into all cell types found in the adults. In these organisms, the first cleavage is asymmetrical, with the two blastomers receiving different cytoplasmic determinants. However, even in species that have cytoplasmic determinants, the first cleavage may occur along an axis that produces two identical blastomers, which will have equal developmental potential. This is what happens in amphibians, for instance, as demonstrated in 1938 in an experiment by German zoologists Hans Zippermann. Thus, the fates of embryonic cells can be affected not only by the distribution of cytoplasmic determinants, but also by how their distribution is affected by zoologists' characteristic pattern of cleavage. In contrast to many other animals, the cells of mammalian embryo remain totipotent until the 16 cell stage, when they become arranged into the precursors of the trophoblast and inner cell mass of the blastocyst. At this time, their different locations determine their phase. The early blastomeres of mammals seem to receive equivalent amounts of cytoplasmic components from the egg. Indeed, up to the 8th cell stage, the blastomeres of mammalian embryo all look alike, and each can form a complete embryo if isolated. Regardless of how similar or different early embryonic cells are in a particular species, the progressive destruction of a potency is a general feature of development in all animals. In some species, the cells of the early gastrula retain the capacity to give rise to more than one kind of cell, though they have lost their totipotency if left alone. The dorsal ectoderm of an early amphibian gastrula will develop into a neural plate above the notochord. If the dorsal ectoderm is experimentally replaced with ectoderm from some other location is in the same gastrula, the transplanted tissue will form a neural plate. If the same experiment is performed in late-stage gastrula, however, the transplanted ectoderm will not respond to its new location and will not form a neural plate. In general, the tissue-specific fates of cells in a late gastrula are fixed. Even when they are manipulated experimentally, these cells usually give rise to the same types of cell as in the normal embryo, indicating that their fate is already sealed. Cell fate determination and pattern formation by inductive signals. Once embryonic cell division creates cells that differ from each other, the cells begin to influence each other's fates by induction at the molecular level. The effects of induction, the response to an inductive signal, is usually the switching on of a set of genes that make the receiving cells differentiate into a specific tissue. The role of induction involves the development in the nematode C. elegans. Here we examine two other examples of induction and essential process in the development of many tissues in most animals. The organizer of spearmint and mangold. The importance of induction during development of amphibians was dramatically demonstrated in transplantation experiments performed by Hans Spearman and his student Hailed Mangold in the 1920, based on the results of their most famous experiments summarized in this figure. They concluded that the dorsal lip to the blastomere in the early gastrula functions as an organizer of the embryo by initiating a chain of inductions that results in the formation of the notochord, the neural tube, and other organs. Developmental biologists are working intensively to identify the molecular basis of induction by Spearman's organizers, also called the gastrula organizer or simply organizer. An important clue has come from studies of a growth factor called bone morphogenetic protein. For BMP, bone morphogenetic proteins, a family of related proteins. Amphibians BMP4 is active exclusively in cells on the ventral side of gastrula, inducing these cells to travel down the pathway toward formation of ventral structures. One major function of the cells of the organizer seems to be to inactivate BMP4 on the dorsal side of the embryo. By producing proteins that bind to BMP, rendering enable to signal. This in turn allows formation of dorsal structures such as the notochord and neural tube proteins related to BMP4 and its inhibitors. Notice the also directions. are also found in other animals, including invertebrates such as the fruit fly. The molecular suggests that they evolved long ago and may participate in the development of many different organisms. The induction that causes dorsal ectoderm to develop into the neural tube is only one of many cell-cell interactions that transform the three germ layers into organ systems. Many inductions seem to involve a sequence of inductive steps that progressively determine the fate of cells. For example, in the late gastrula of a frog, ectoderm cells destined to become the lenses of the eye receive. Now, notice this. The eyes receive inductive signals from the ectoderm cells that will become the neural plate. Additional inductive signals probably coming from endodermal cells and mesodermal cells. Finally, inductive signals from the optic cup and outgrowth of the developing brain complete the determination of the lens-forming cells. Okay, formation of the vertebrate limb. The action of the gastrula organizer is a classic example of induction, and we can see that the organizer introduces cells to take on their face in appropriate locations relative to each other. Those inductive signals play a major role in pattern formation, the development of an animal's spatial organization, the arrangement of the organs and tissues in their characteristic places in three-dimensional space. The molecular cues that control pattern formation, called positional information, tell a cell where it is with respect to the animal's body axis and help to determine how the cell and its descendants respond to the future molecular signals. A limb bud. Notice here a limb bud consists of a core of mesodermal tissue covered by a layer of ectoderm. Two critical organizer regions in the limb bud have profound effects on the limb's development. These two organizer regions are present in all vertebrate limb buds, including those that will develop into forelimbs such as wings or arms, and those destined to become hind limbs. The cells of these regions secrete proteins that provide key positional information to the other cells of the bud. When also notice also the tissue transplantation experiment outlined as shows in this figure, 3 digits like cells farthest from the ZPA former anterior structures, including the most anterior digit. Also supports the hypothesis, this figure, that the ZPA produces some sort of inductive signal that conveys positional information indicating posterior. Indeed, researchers have discovered that the cells of the ZPA secrete an important protein growth factor called sonic hedgehog. If cells genetically engineered to produce large amount of sonic hedgehog are implanted in the anterior region of normal limb bud, a mirror image limb results, just as if a ZPA had been grafted. Their evidence from studying the mouse version